Okay, so here we are on the second page. You turn to where it says at the top of the second page, multiple alleles. It says for some traits, we'll add in here, there are more than one allele. Okay, so what do I mean by that? I mean different versions. That's what I mean by more than one. Different versions of a gene. Right, so we'll look at it next, but different blood types other than just blood type A or O or B. There's in fact 4 different types: A, B, A, and then B, and then AB, and then O. Type O, by the way, we'll get to that in a second here, is a universal donor. It's rare. You can donate blood to anyone. Okay, so let's take a look at the different blood types due to the 4 different alleles. We have A, B, and then AB, and then O, the rarest. We have here the genotypes that result in these blood types. We have can have a combination of the allele A and I, or IA, IA. You know what? I'll use a different color. Okay, so for B, we have 2 possible genotypes, because IB is dominant. You can have IB and little I, or 2 dominant. Okay, for blood type A, there's only one genotype, IA, IB. And these little i's I've been talking about Two of them together give us the type O blood, the universal donor. Okay. Next, epistasis. Epistasis is just, we go back here, it's another type of dihybrid cross, except it's not going to end up in the ratio that we expect, where it's 9, 3, 3, 1. You'll end up in a, works out to be a 9, I believe it's a 9, 3, 4. Anyways, it's hard to see here, but you've got basically what is epistasis, and I'm running out of room to define it here, but epistasis, if we go over here, I'll define it. It's a gene at one location okay, when a gene at one location affects the expression of a gene at another. Okay, so an example of that is what we see here with the mouse. So in general, you can have either a black or a brown colored mouse, but in some cases you'll end up with an albino or a white colored mouse. Now, why is that? It's because of this epistasis. There's a gene at one location that affects in this case the expression of the brown and the black alleles. Okay, so if you look here, big B here codes for black and it's dominant. Little b codes for brown, it's recessive, but neither black or brown will be exhibited if you get the following. You get a lowercase c, lowercase c. You get a combination of a big C, little c, or a big C, then you'll get the expression of either the black or the brown. So let's take a look at this dihybrid cross here. You'll get any of these that I'm going to code in yellow. You're going to get the expression of either black or brown. So there's a big C there, big C there, big C, little c, big C, little c. Okay, you'll get either, in this case, you're going to get all black, but the fact that there's at least one uppercase C means that you'll either get black, as in this case, or brown. Okay, more examples of that are here. Big C, you're going to get black here. Big C, you're going to get brown because there's two recessive little b's. Big C, little c, black's going to come through. Big C, little c, brown's going to come through because you've got two recessive little b's. Okay, etc. Okay, I'm underlining all the ones where a black or a brown could occur. Okay, where does the black or the brown not show up? Although hypothetically they could. It's in these cases here. I'm going to underline in red. In this case here, there's a little c, little c. Doesn't matter what preceded it. The mouse turns out white. Little c, little c, little c, little c, little c, little c. Okay. So in these four cases, you'll end up with a white mouse. Because of this understanding of epistasis, where the little c are the set of genes at one location that affects the expression of the other brown and black genes. Okay, so your ratio here is, it turns out to be 9 of the gray or black, and then 3 of the brown and 4 of the white. Okay, rather than what you'd expect, what you should expect with a dihybrid cross is 9, 3, 3, 1. Instead, you get the 9, 3, and the 4. Okay, on to polygenic. Polygenic inheritance. Some traits show additive effects. Of 2 or more genes. Okay, you just have to know that definition. Okay, an example of that would be skin color. In humans. Okay, next, sex-linked inheritance. As a general rule, a male always inherits a sex-linked trait. Of 2 or more genes. Yep, sorry, made a mistake here. Sex-linked trait from male always inherits a sex-linked trait from the female parent. Okay. So an example of this would be color blindness. Red-green color blindness is linked to the X chromosome, the female chromosome. Let's just remind ourselves, when we inherit XX for the sex chromosomes, we get a female. When we inherit XY, we get a male. Okay, so let's look at, we're going to draw a Punnett square here. C will represent, big C will represent not colorblind. and little c colorblind. Let's see what happens when we mix a carrier. Let's see here, we're going to pick up the Punnett square. I need more room. Okay, see, I have a female carrier. It doesn't show the traits because she's got a capital C here. But she's a carrier because she's got a lowercase here. Okay, then we'll cross that with the male who's colorblind. Okay, so the first cross we get X, big C, X, little c, a carrier but not colorblind female. You need two lowercase for the females. Cross here, you get X, capital C, and then Y, a non-colorblind male, and then here you get X, lowercase c, X, lowercase c, that's a colorblind female, and then here you get the lowercase c there, and a Y, that's a male who's colorblind. Okay, let's go on here. Let's read what it says. Make this smaller. Okay, so next, linked genes, what are they? They're traits that are inherited together. So how is that possible? It's possible because these genes are actually physically linked. So if you take a look at what we see here, let me check here. I just circled it. We've got an example here of linked genes. Physically, they're linked together. It says here it's a set of genes at different loci, different locations, on the same gene that except for crossing over, tend to be inherited together. So these two, in this case, three linked genes physically tend to stay together unless crossing over occurs. Okay? So unlinked genes, it says here on the left, are located on different chromosomes. So just for clarity's sake, loci, that is location on a gene. Loci means location on a gene. Okay, pedigrees. What is it? It's a visual chart visual chart that shows family history. Or traits that are passed down. They can be interesting to view and can be important tools in determining the pattern of inheritance of specific traits. Okay, uses of pedigrees. They're used primarily by genetic counselors. When couples decide to have children when there's evidence of a genetically inherited disorder in one or both families. Pedigrees are also used when trying to determine if someone is carrying hereditary disease. Okay, so here's an example of a pedigree picture. These Roman numerals here, they indicate the number of generations. The squares are used to indicate males. And the circles, females. Okay, if the individual is affected by the trait, we darken the shape. A line between the male and female indicates a marriage. So this is a marriage, 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 marriage. Okay. Right in here, marriage, or some sort of union. It should be a marriage. Anyways, it says here a line drawn down from the marriage line or a union indicates offspring. So that would be, that's going to indicate or lead to offspring. It spans out in either direction. Okay. Because there's more than one offspring in that case. Line down, there's the offspring. Line down to the left and to the right, two offspring. Line down, one offspring. Okay. So a line again that's drawn down from the marriage line vertically. The yellow lines there that I drew indicate the offspring. Sometimes you'll see some shapes filled in only halfway. And this notation indicates a hybrid or carrier of the trait. Okay. Third page, analyzing simple pedigrees. Let's read through that. Says here, a pedigree is just like a family. It's like a family tree, except that it focuses on a specific genetic trait. A pedigree usually only shows the phenotype of each family member. Okay, that basically what family member exhibits physically. With a little thought and the hints below, you might be able to determine the genotype of each family member as well. Okay, so in some cases. So let's look at some of these hints. It says, and let's see if I gotta highlight some of these. Ooh, I can do that. It says here, we're going down the list here where it says hints for analyzing. I'm going to make it thick. Okay. So, if the individual is homozygous recessive, then both parents must have at least one recessive. Oops. Both parents must have at least one recessive allele. Right? Number two, if an individual shows the dominant trait, then at least one of the parents must have a dominant phenotype. Okay. And then thirdly, if both parents are homozygous recessive, then all the offspring will be homozygous recessive. Okay, so let's go on to where it says early genetic engineering, selective breeding has produced domestic animals such as dogs, horses, cats, and farm animals, as well as most food crops. Farmers, breeders, and scientists select desirable traits that are passed on to the next generation. They're breeding, obviously, in order to pass on those traits that are desirable. Luther Burbank used the process of hybridization to combine disease-resistant potatoes with potatoes that were good for eating, and as a result produce plants with both characteristics. Inbreeding, I'm sure you've heard of this. Inbreeding is used, it's actually helpful in some cases. Inbreeding is used by breeders to maintain unique characteristics. However, there is an increase in the chances of a cross between two individuals that carry recessive alleles. For a genetic defect. It can go unnoticed until two recessive alleles are inherited, and that's when you see the defect. Scientists increase genetic diversity. You can force genetic diversity. You can force it to happen by inducing mutations. In the DNA by using X-rays. That's why X-rays have to be limited, or absorption of them have to be limited, because too much of them can cause mutations. So they introduce X-rays, and chemicals can cause mutations as well. Toxic chemicals. Mutations occur spontaneously in nature. So mutations occur randomly in nature for various reasons. But that happens at a very slow rate. So scientists speed it up. How do they speed it up? Again, by adding these X-rays or chemicals. Most mutations are harmful. Or neutral, but every once in a while we get a desirable trait. Okay, back side of the third page, where it says applications of genetic engineering. We're almost done here. Uses for blank organisms. What organisms am I going to be talking about? Transgenic engineered organisms. Transgenic engineered organisms, they can produce useful products for us. For example, number one, it says transgenic bacteria can produce useful products such as human proteins. Okay? So how can they produce human proteins? Because they contain genes from us. We've actually put some human genes in bacteria, and as a result, they can produce proteins that we need. That's what a transgenic engineered organism is. It's one that contains genes from another organism. Two, transgenic animals that contain human genes can be used as models for human organ systems. The genes that are inserted make them more human-like, so that when we test a certain drug, we can more accurately see possible effects in a real human. Okay, because the animals take on characteristics in their organs or their organ systems that are similar to ours. We can do tests on them to see how the different drugs perform, and then get an idea of whether they're going to affect human beings in a positive way or not. Three, transgenic animals can also be used to produce human proteins to be used as drugs. So the animals can also produce human proteins. Sheep and goats are currently used for this purpose. Number four, transgenic plants have many potential pharmaceutical uses as well as for food. Okay. Genetically modified foods genetically modified foods, or GMOs, can be engineered to be disease ridden. Not disease ridden, but disease resistant. Or to produce natural pesticides. Cloning. A member of a population of genetically identical organisms. Okay? The organisms are clones because they're identical genetically, through and through. They're copies of the original. While we have been able to clone many organisms for years, it was thought impossible to clone a mammal. From an adult cell. Dolly, the sheep, was the first mammal ever cloned in this manner. Here is the basic procedure that they followed. An egg cell is harvested from a black-faced ewe. And the nucleus is removed. A donor cell with the nucleus is taken from a white-faced ewe, and the egg and the donor cell are fused. They're fused together with an electric shock. The fused cell begins to divide. And that's what the whole purpose of the electric shock is. That's what allows the cell to divide and then form an embryo. The embryo is placed in the black-faced ewe foster mother, and when the baby is born, it will be a white-faced lamb. Okay, so we're done with the notes. This is all practice I'm going to mention in class. That'll be an assignment.